The experiment we conducted involves a strain of algae called Chlamydomonas reinhardtii and the use of P49 medium dilution. The people that conducted this experiment are Evan Ostrander, Timothy Barr, and Robert Venema.
So our hypothesis was if we dilute Chlamydomonas reinhardtii in the P49 medium using one sample by 1/2 and another sample by 2/3, then the algae cultures that are diluted will increase in fatty acid levels the most after 2 weeks of growing because the dilution of the nutrients will stress the algae in order to produce a higher lipid content.
For our experimental design, we used 3 different samples. One was a control sample, the other one was diluted with 1/2 medium, and the third was diluted by 2/3 medium. We grew our base cultures for 1 week under normal levels, then transferred into the new modified medium after 1 week of growing and grew that for 2 weeks. The first sample was our control. The second one had 17.5 milliliters of algae and 17.5 milliliters of P49 medium. The third had 23 milliliters of P49 medium and only 12 milliliters of algae.
As for testing our hypothesis, one of the many ways that we measured our data was through a spectrophotometer, which is an instrument which measures the amount of light of a specified wavelength passing through a given matter. We measured our algae cultures, all 3 of them, every other day to obtain the best results. Our results showed that on average, our 1/2 dilution sample grew the healthiest out of the given 3 samples of control, 1/2 dilution, and 3/4 medium dilution. Some of the data here seen is the spec data. We have the control in blue, the 1/2 in red, and the 1/3 dilution in green. The x-axis is labeled for the 5 days of the week that we tested this, and the y-axis is labeled as the light percentages and optical density through the spectrophotometer. As you can see, the control and the 1/2 dilutions actually stay relatively the same throughout the 5 days, but the 1/3 and 2 dilution actually raises on the 3rd day. We believe this is because the algae began to die off, allowing more light to pass through on the spectrophotometer, and the culture began to deteriorate.
As our testing continued, we used flow cytometry, which is a laser-based technology used by suspending cells in a stream of fluid and passing them by an electronic detection apparatus. It uses chemical characteristics to differentiate between the different types of cells. It can add up to thousands of particles per second. And our sample with the highest lipid content, according to the flow cytometer, was our 1/2 dilution sample. We gathered our data on FlowJo, and as you can see, the line across each sample is the gated part. And so our first sample on the top left is our control sample, which was 71.6%. Our top right sample was our 2/3 dilution sample, which was 73.3%. And our bottom sample, which was our 1/2 sample, was 87.3%.
So for our third test, we used a Bradford assay. This tests the concentration of protein in a solution. It works by inserting a dye, which then bonds to the protein, and then you stick it in the machine and it measures the amount of bonded dye present. For our sample, again, it was the 1/2 dilution that had the highest levels of protein. This is our Bradford assay data graph. Starting with the bottom left blue square is our control sample. Next up is the 2/3 dilution. Following that is the 1/2 dilution. Our fourth dot is the projected amount of protein we think if it would keep on growing after the 2-week cycle had ended. For our fourth and final test, we PCR'd our samples to see exactly what was in them. When we got the sequencing data back, we learned that it was inconclusive due to a mechanical error. Not everything in there was algae. We even got some mice DNA in the sequencing data. We don't know how that happened, but it was probably due to a mechanical error.
Some of the materials we used are a microscope, which you can see a picture of our Chlamydomonas reinhardtii culture in the bottom right, the 35 milliliter flask, which you use in a picture in the top right, with our control, our N1 representing the 1/2 dilution, and our N2 representing the 2/3 dilutions. We also used a flow cytometer you could have seen on previous slides to represent our data, our spectrophotometer, the Chlamydomonas reinhardtii cultures, of course, and P49 medium. We used about approximately 40 milliliters of that. We also used a centrifuge to conduct some of our DNA sampling. We used slides, measuring scales, a thermometer, and an orbital shaker to conduct all of our experiments.
So in conclusion, our data supports our original hypothesis that 1/2 and 2/3 of a P49 medium dilution will do the best overall for our algae samples. And overall, the half dilution sample had the best increase in protein, lipids, and overall growth. If we could redo this over again, we would probably add a fourth sample, which would have been 3/4 of P49 medium dilution. And some possible errors that may have occurred through this experiment may include, but are not limited to, improper measurements, contaminated flasks, mistakes in reading data, and unseen contamination. If more caution and precision was used in future experiments, the results may change drastically for better or worse.